Hi, everyone. My name is Martin. I'm talking to you from the Genome Sciences Center in Vancouver. I know there's lots of people joining, and everybody's from a large number of different backgrounds. And you might think of whether this will be relevant to you. Rest in mind that I want to make this as relevant to everybody as possible. And even though I work in biology and genomics, and some of the examples will be from those fields, I will try to underscore some of the ideas and concepts that will make this broadly applicable. If anybody has any questions, please feel free to send Jana an email at any time during the talk. I can stop and address them as well as I can. I want you to keep in mind that as scientists and as broadly people who are representing science and working in science, one of our most important responsibilities is to explain things well. It's to explain complex things well, simply at first, and then build up the complexity of our explanation as the audience is ready for it. This means choosing productive metaphors. This means using words and ideas and similes and all of these tools of explanation that do not unnecessarily undermine the complexity of what we are eventually wanting to talk about. I've put up this question because it's an example of I think, a failure on the part of the scientists, not on the part of the person asking the question, to explain ourselves well. You may have seen this question, and to many of you, this question may not make much sense. But if you imagine where the person asking this question obtained their information from, for example, Google Images, they might have found something like this. And this is a pretty common trope about evolution, and you may see this tiling the Google Images result page. Now, in light of this particular image, that question makes complete sense because this image does not show some of the fundamental ideas of evolution, like speciation and extinction, and even the fact that this process winds up looking much more of like a tree than a single timeline. And when I did this, you in fact had to scroll past this image to finally get to an image that looked something like a tree, which is a really beautiful image, and it needs to be further up. We need to work a lot harder as scientists to try to get productive explanations. They can be accessible and productive at the same time, closer to the forefront of people's minds. When we fail at this, what can happen is that the visuals and the explanations that we produce, and for a moment there, I don't want you to just think about visual explanations. I want you to think about verbal explanations, written explanations, all sorts of explanations, may be indistinguishable from junk science. For example, here is an image on the left of a so-called synastry chart, which is an astrological mechanism to try to discover the compatibility between two people. And on the right, something about probably gene expression. And if you were to look at these at face value, the thing on the left seems to have a lot more accessible information and actionable information. There's numbers, there's symbols, there's some kind of connections, they're placed in some kind of a rational way, presumably, or at least according to some rules. Whereas the thing on the right doesn't seem to have any actionable information in it whatsoever. And what can happen when we create images like the things on the right? Even though they may be based on fantastic science, they may be based on very useful models and part of a very useful conversation in how to advance understanding, if they don't do their job in explaining, then for a large number of people, they're actually quite useless. So I want to come back to this idea of visual metaphors, because I want you to be thinking about whether the thing that you're drawing or saying or writing is furthering that explanation or is getting in the way of actually a robust understanding of what it is that you're trying to describe. So the first quote here by Feynman is one of the most wonderful and I think very useful metaphors of science. This idea that we're kind of at this fulcrum of this balance. Inside of us is indeed a universe of atoms. There are many, many particles that we are made up of. And at the same time, beyond us is an entire universe made up of such particles, and we could see ourselves as individual particles in that universe. This speaks to scale. This speaks to the fact that matter is made up of little pieces. And Feynman was once asked, what would there be a sentence that you would want to communicate to our descendants or to other people that embodies the knowledge that we have about physical reality? And he came up with something about that matter is made out of small particles which attract each other at great distances, but when they get close, they repel each other. Kind of captures quite a lot of physical science and how we understand the world works. Now, the second metaphor on the screen from nature, and not just from nature, but many people use this term, the fact that genomics is a blueprint, it is, I think, very unproductive, and I think really gets in the way of communicating what it is that the genome does and how it works. And I just want you to consider this for a moment, because we'll connect these bad metaphors to visual representations later, and try to tweak the visual representation so that it doesn't distract the reader and it gives the reader the most number of productive ideas that they can have about the information or the concepts at any given time, which is your goal. So the question is, does talking about the genome like a blueprint give the audience the largest amount of productive thoughts? It's actually very difficult to have a meaningful conversation about the genome while calling it a blueprint. So if you think about what a blueprint is, and if you think about what a genome is, you realize that, oh, well, obviously, they're very, very different. But they're fundamentally different in that the blueprint shows you what the thing is going to look like. The genome does not show you what the thing is going to look like. In fact, the phenotype, the appearance of the organism, and the genotype, its genome, are not at all easily connected, and we do not know how to do this yet for most things. If you give me some genome, the only way I could figure out what this genome is of is if I could try to find a similar genome, or if I could find a level of similarity between this genome and other genomes. In other words, I need other organisms, other genomes to figure out what this one does. I don't need other blueprints to figure out what this blueprint does. This blueprint is very, very obvious. The blueprint also probably suggests what the thing is made up of, whereas the genome does not. The genome does not encode the substrate that the organism is made out of at all. What does the genome encode? So if you think about this idea that your home has a genome. If your home had a genome, it certainly wouldn't be a blueprint. What would your home's genome look like? It would look like this. These are a bunch of tools used to make the home. The thing that the home is made out of is not in the genome. The workers who may build the home or who use the tools are not in the genome. And what makes this complicated when you think about when we're studying genomes and why it can sometimes be slow going, is because we need to figure out what each of these tools does, and it may not be that obvious. I mean, you may know what a hammer does, but unless you're excellent at constructions, I mean, so maybe some of these things kind of confuse you. Moreover, if you come across a genome that's slightly different, like this one, even spotting the differences itself might be challenging. It isn't at all obvious what the consequences of these differences are. It may be that that changed angle in that right angle ruler has an effect on the home, or it doesn't, because there's other tools that make up for its deficiency. It could be that the home is still standing, except maybe just some corners are off. It may be that the home falls down, and it can never stand. It's not viable. You just don't know. You actually have to understand how the entire thing is put together before you can understand what the impact of any one of the tools is. And this is what makes discovering why changes in the genome are what makes discovering how they affect the organism really complicated. And if I think about the genome as a blueprint, I can't have this conversation at all with anybody. I have to, at some point, say, well, it's not exactly like a blueprint. And so if it's not exactly like a blueprint, you should never call it a blueprint. The way you explain things verbally or on the page, there are some guidelines. There are some long-standing guidelines about how to phrase your words, how to pick your words, how to construct paragraphs, and all these kinds of things towards a productive and clear explanation. And some of you may have read this book, which is now fairly old and a little stodgy, but I think still very useful to flip through. And it translates into the idea of visual style, in that you can apply some of the rules of grammar, some of the ways in which we constrain how we speak and write, to how we draw. And we're not born articulate speakers. That can take a long time to sort out. One of the ways in which we can get there faster is by, in a way, listening to ourselves and asking ourselves whether we've taken the shortest path to clarity, unless we're trying to be poetic, in which case, have we succeeded at being poetic? Oftentimes we fail at both, and then you self-correct and you try better next time. For visual style, it's very much the same thing. You have to look at what you've drawn and try to act the role of the objective observer and ask yourself, have I done a good job in explaining? And even though you're seeped in the concepts and the data, and of course it makes lots of sense to you, and you don't want to let go of any of these hard-won details that you've worked long hours for, you do have to stand back from it and imagine a blank mind and how it might perceive what you're showing it. And there is a hierarchy of importance that you have to advance forward. And you don't want to jump into the tangents before you've determined the main trunk or the main subject of your talk. What does visual style kind of translate to? What does that look like? Well, it looks like this. These two panels show the same objects, but they show them in a different way. Visually, I have chosen different words, and I have chosen a different emphasis, and I've used simple words, and I've used small words, in that there is no decoration, there's no garnish, like gradients, or strange shapes, or unsymmetrical curves. The idea is to reduce the number of degrees of freedom or the number of visual options presented to the reader. Because when you have a large number of these options, you can get into trouble where the artistic freedom you've taken in representing your elements can actually get in the way of explaining and get in the way of connecting to features about the data that you want to highlight. For example, if on the left and in the middle, you look at the purple A and the D element, The purple A element has a DA inside it, which is labeling the element, and it has a star above it. But the purple D element has the label above it and the star inside it. And you wonder why. Is that meaningful? Or is that just because the D doesn't fit into that purple oval? And so the D has to be outside. So why is the D oval so small? Why couldn't it be bigger, like the E oval? Is the D element actually smaller? Surely it must be, because then why would you make it so small that the label wouldn't fit? But then if that's the case, then you start to think, well, then is B and E the same size? Is C bigger? Is the size on the screen of these elements exactly proportional to their size in physical reality? You start to have all these questions about proportionality and about whether this is drawn to scale. And you start to wonder, well, if the size matters, does the shape matter? Why do they have different shapes? And you have all these questions, and none of them are answered. And one of the things about drawing data is that you want to reduce the number of irrelevant questions. If these issues are important, then you should encode them as clearly as possible so that it's obvious that you're doing this with intent. Otherwise, you should back off and try to use the simplest possible representation. And kind of as we get into examples, it's important to think about how to approach it. And I know that because most of the time I think we spend more time talking than writing and more time writing than drawing. Sometimes maybe some of us spend more time writing than talking, I don't know. And how do you approach this idea of drawing things? I think the only way to do it consistently that produces good results is to take kind of a top-down view where you want to achieve certain things, like you want to limit redundancy, you want to achieve consistency, and you want to be clear. And then you ask yourself, how do I use the tools at my disposal? How do you use these visual elements like symbols and colors and arrows and so on to achieve that? And whatever shapes and colors and alignment approaches I use have to satisfy this desire for things like salience and relevance. The thing that stands out on the page is the thing that is relevant. Otherwise, I have misplaced my emphasis. You should not be thinking about, I have to draw a scatter plot. I will force my explanation to match what the scatter plot produces. You should not think about, I have to draw a triangle, and regardless of what it is that that triangle represents. Nobody thinks that. And yet, you often wind up being constrained to use the tools that you know, which produce the things that you know how to produce, like a scatter plot. I know how to make a scatter plot in R, so I'm going to make a scatter plot because I don't know how to make another kind of plot. And we're, in a way, always constrained by that. We're limited to use the words we know. We can't use the words we don't know. You could try to learn new words and then have them waiting in the back, waiting until such time as they could be deployed well. And that takes a lot of effort. So I realize that you may sometimes be looking for the right word and maybe using slightly not the perfect word for it. You might be looking for the right plot or the right drawing tool or the right approach, but be unfamiliar with it. And that's why looking at how other people have done things and say, well, that looks like a good solution, that looks like a good word, can I use that, is really important. Keep your eye out on it for it. A lot of the things that I'll be talking about and the things that I've already talked about are written up in our points of view column in Nature Methods, which is a long-running series of brief vignettes about drawing data and thinking about visual representation. And even though it is in the field of biology, even if you're not in the field of biology, I encourage you to read it because it's eminently translatable to other fields. There are ways in which we look at and perceive and organize information that's fairly universal. And I can count on you to look in one part of the screen and not the other. Regardless of what the left side of the screen might be yelling at you, you're going to look on the right because of all that negative space. And this idea of emphasis achieved by emptiness is really important, and I want you to think about it when you come to the point in your figure making or in your drawing process where you need to achieve some amount of emphasis or focus. It isn't always going to be productive to use red or bold or drop an arrow in there. Sometimes isolating something from its neighbors is enough. And in fact, often that is the right solution. You probably have seen these kinds of title slides or slides in general where there's a lot of elements, and many of them are incompatible with one another with respect to coloring, and there's a lot of things that compete for your attention. And the thing that this is about, the main point, isn't the thing that you see first. The logos at the top with the white background and the dark slide background actually really interfere with things. This tremendously wonderful red image of the brain that looks very much like it's not a normal brain because it's red is kind of offset by this really dull scan on the right, on the left, the gray one. And it loses its emphasis. And I would suggest that you try to have a hierarchy of importance. What is it that is important on this slide? Are the logos really that important? Well, they're important, but they should be subordinate to, let's say, the title. And by actually taking the title outside of a box and giving lots of negative space around it, you focus the reader's attention on it. And then you keep that one image that really speaks to this idea of maybe the unhealthy brain or brain on fire or whatever connection you want to make with it. And that is the only splash of color on the page. And that image works much better, and it doesn't get lost. It has something to offer, and it is able to offer everything to the reader. Because there's no competing elements. There's this idea that we think that when we explain, or we think that when we draw things, we have to follow some kind of a process. We have to follow some kind of a series of steps which are prescribed to us. Now, yes, there are guidelines, and we should all be mindful of those, but we should not be sticking to this prescribed process too firmly. Software might be telling you that you might need to click to add title. And so you feel like, well, I need a title because the software is telling me that I need a title. Everybody has to have a title. Don't we have a title? Don't you have to say what it is that you're talking about on the slide? What happens if I put up an image and there's no title? How will people know what I'm talking about? So you wind up with slides like this, which are very compelling. And remember, there's a speaker that's accompanying these slides who's at the front of the room, or maybe on a tiny little chat window box like this one, talking to you about what it is that you're seeing. So this person has the opportunity to provide the foreground, the verbal foreground, to this visual background. And these images are compelling, but they're not as compelling as they could be. And the reason why they're not as compelling as they could be is because the bloody text gets in the way. You read the text, and it takes you away from the purely analog experience of imagining what these environments are like. And as soon as I take the text away, I think the experience is totally different. I think it's much richer. And remember, this is the point where I talk about population growth and urbanization and overcrowding. So I say those words, and you don't have to read them. And I talk about, you know, polluted water and so on. So I want you to think about whether all the elements that you're putting down on your slides, your posters, your figures, are actually necessary, in that are they a crutch to you to remember what to say if it's on a slide, or are they words that are somehow indispensable and are not implied by the image? I also want you to use your imagination more. We're scientists. We're supposed to be people who think about things and push against this fog of confusion. And I want you to play a little bit and inspire your audience a little bit more than you are already doing. Scientific presentations are too often too dull, too conservative, and completely lack the human aspect of how science is done, which is people getting frustrated at the fact that they're confused, people imagining a better world where some of these problems are solved without getting all too dramatic about it. So instead of having a slide like this, where, you know, you put up a land snail, which is what the land snail looks like, and it's exactly what a snail looks like in people's minds without even them ever seen a snail. You know, nothing about this representation of the snail really inspires my imagination. Try to put up something surprising. Try to put up something delightful and say, look at all of these wonderful patterns, but never say that. Never say, look at these patterns. Just leave them up there and let people enjoy the complexity and the colors and get them into a mood of feeling and thinking, and leave it up for a while and then move on and start your talk. There are these guidelines. So I did say that you shouldn't be following these rigid rules that are prescribed by software. Sure. But there are some rigid rules about rigidity that you should definitely abide by until such time as you are ready to let go of them. So you should always know what rules you're breaking. If you don't know what rules you're breaking, then you can get into trouble. You should always align and space things evenly and ruthlessly, and you should be looking for things that are out of alignment on your slides and your figures and everywhere around you as much as possible. And the reason for that is that you provide the eye a grid system, you reduce the amount of freedom for the placement and size of elements, and you focus on changes that are driven by data and not by formatting. So if I were to say, how many dots do you see? You're still counting. But if I arrange them nicely, you don't even have to count. You can kind of pre-tentatively judge how many dots there are. And the only reason why you can do that is because they're on a grid. Your mind sees this pattern and is able to immediately extrapolate to all the different positions, whereas before there was no pattern. So when you draw things that try to explain patterns, and you use elements to explain those patterns that are not equally sized or that are not equally spaced, then you leave some confusion in the mind of the reader about whether the spacing and the sizing is actually an important part of the pattern or it's not. Now, the thing on the left, I don't know whether it's relevant that the sizes and positions of things are different. And the thing on the right, it's obvious that they're not, because everything is rigorously aligned and placed on a grid and all the elements are the same size. And because we know that in nature not all the elements are the same size, if these are exons, for example, then we realize that this is a schematic in which there are certain things that have been abstracted and simplified for the purpose of elevating the visual importance of the pattern. I'm not implying that the exons are all the same size, nor would anybody think that. This idea of alignment is really important because it actually helps you put things together and reduces the panoply of choices that you have when you set out to draw something. And drawing data isn't just about plugging the comma delimited file into an app and getting a scatterplot out of it, because that part of it is actually relatively easy. It's organizing everything on the page in some kind of a flow and in some kind of a grid that relates relevant information together, and it helps you make judgments across different panels. That's harder, and there is no software that can help you do that. So, in this figure, what are the issues here? There's a bunch of different issues. So one of them is that things are not well aligned. The text size is too large. The data actually themselves are drawn with very small symbols. There's a lot of things that are competing for your attention. There's some inconsistencies with coloring. For example, in part D, the white bar is the no stain bar, but in part E, the white bar is the IgG1 bar. And so you have the same color used to represent different things. How would you, we're going to back up and start this from the beginning. You would say, well, I need 5 panels. So I would lay the panel labels down, and I would put my axes down. And the axes would be aligned, and it would be light. And at this point, I should not have yet overwhelmed the page with information because no information has been shown. Then I would draw the data, and I would make sure that the size of the data points and the size of the panels and the white space around them is balanced so that there's enough clarity and separation between the data points, there's enough separation between the panels, and all the elements can be clearly seen and there's not occlusion. And here, you might feel like, well, those individual data points could be bigger. Sure, they could be bigger. Make them bigger. Think about, are they big enough? Are they too small? Should I maybe use different tone of gray for some of the scatter plots with some of the points with the error bars? Sure. As long as you're thinking about that, chances are higher that you're going to wind up with a better solution. Then you can drop your images, then you can drop the final labels in, and at no point in time are any of the things that you drop in going to overwhelm the data that you've already drawn. They're annotations. And the typeface should be light, and everything should be aligned. And one of the ways in which you can check whether you've done a good job is if you only show one of the layers in the image to yourself, for example, the labels, and you look at them and you look for things that are out of place, you look for things that are not aligned, you look for inconsistencies, and so on. And these labels by themselves should already give you an idea of where the rest of the elements get placed. In other words, each of the different layers acts as a kind of reinforcement for the organization of other layers. And then when you have a large number of such layers, everything seems to lock into place. It's a bit like a jigsaw puzzle. So it's one kind of improvement that you can make. Here's another such figure where maybe you can think about, well, how do I apply this process from the previous one to this one? You might say, well, I don't know anything about what this figure is showing. That's okay. Nobody knows anything about what this figure is showing. It isn't about what it's necessarily showing, but there are certain very easy fixes that you could do to bring out some of the issues about what the figure might or might not be showing, or even just to explain things better. And the idea is to try to reduce the number of colors, really make a much better connection between what is shown on the left and what is shown on the right. Clearly label all of the stages. There's 6 stages here in this transformation process. By making the labels larger and by carrying through the entire figure this idea of the red, which is DNA, and making the red really salient by not using any other colors except for that splash of blue, you can follow that trail of red. It's much harder to follow the trail of red on the left. And then you can also look at whether the symbols you've drawn are rationally placed. If you look at just the arrows and some of the labels in the original figure, which I show here on the left, they're a jumble. The arrows are pointing in a variety of directions. Stuff in the middle doesn't seem to suggest where the original elements were drawn. Whereas with the redesign on the right, you have a much better flow of things. And even though there's some curved arrows in that transformation process in the middle of the screen, you have an idea of where the other elements should go. And the idea of alignment, I keep talking about it because it's one of those really easy things to fix, because every software package, be it Keynote or PowerPoint or Illustrator or whatever, has an alignment palette. And that palette offers a series of tools: to left align, to center align, vertical align, space evenly, and so on, distribute, and all that. And it automates all of that for you. And you should be always using those to put at the same vertical or horizontal position things that have the same meaning. So, for example, in this image, you could argue that this CB1 element, this orange element, is important. So, and if it's important, then it would make sense to try to perhaps center the image on that element. And the neo element is removed, as you could see from the label at the bottom, genomic integration after removal of neo. But if I were to remove neo from the top element, you could just kind of imagine that bit on the right that's labeled 3' genomic just sliding in towards the left, and that would be the that new element removed. But if you look at the image below that actually shows you what has happened, it looks quite different. It's been shifted to the right. There's an HA element that's now kind of appeared that wasn't there before. So this idea of how to represent something once a little bit of it has been removed should be done with more consistency. So I would suggest to draw it in this manner, where you center everything on CB1 and you provide the vertical axis as the axis of detail. So the things at the top are the things that are most detailed. And you can flip this around and you can have that at the bottom if you wish, where you have this sequence. And then those gray funnels bring you back to a broader context. And then right in the center, you have of the image, which is where the eye goes. You have the thing that the image is talking about, which is this targeting vector. And then below, you have the targeting vector without the green element. You don't even have to say removal of neo R1, because it's really obvious that that thing has disappeared, because everything else has stayed the same. And also notice here that we'll come back to color later, that color is a very powerful way to achieve focus and to draw people's attention. And the more colors you use, obviously, it dilutes the effect of all the previous colors you've already deployed. In this case, the genomic green boxes are the background boxes. That's just part of the genome that we're not particularly interested in talking about right now. So they should be a background color. They should be a background tone, which means gray or white if you want to just have an outline. There's really no need for green here because now in the original figure I have one more color that I need to deal with. By making things gray, gray is the synonym for background. It's the control, so to speak. And people will not have gray overwhelm their attention before they've looked at other colors. This is a really important point that's difficult for biologists to actually implement, this idea of the spherical cow or radius r, which is a physics joke. Because in physical science, you can approximate your system, your model with the first term or the first two terms. There is a workable approximation to the system that you're studying. In biology, that's much less the case because there is no workable zero-order approximation to a cell. You need almost a ton of complexity to just get the cell, and the cell can't be broken down any further. It's kind of irreducible in that way. You need all the parts to make the cell. Whereas in other fields, you can neglect certain pressures on the system or certain behaviors of the system for the purpose of modeling and for the purpose of just getting a handle on how things work. But that doesn't mean that you can't imagine what that spherical cow might be, or you can't aim to have as simple an explanation as possible that captures the essence. So remember that, yes, while the cell might be very complex, you don't have to explain the entire cell to explain one particular part of it. And you can ignore everything else that's going on. And that means not show it, which means using the simplest representations and only those to explain what you want to say. So here's an example of translocation in a chromosome where part flips over and parts of different genes are brought into adjacency with one another, and you get these so-called fusions. So is there a simpler way to explain this? Can this be shown with fewer elements? How would you think about that? You might count and say, well, I have 4 arrows here. Do I need all these arrows? Do I need this gray centromere in the center? Is that important? Is that just getting in the way? How much of the structure of the chromosome, how much of the shape of the chromosome do I need to show to say chromosome? Really, it's just a line, and I'm flipping a part of the line around. This is one option, is you can only have one arrow. The curved arrow represents kind of motion or suggests a rotation together with that tilted element at the top. You don't have to show the before, the during, and the after. You could just kind of show the midway process as the element is flipping and then the after. You can go further and reduce the complexity of the labels. Genes don't have to have 3-letter names. They could have 1-letter names. They could have any names that you want. There's a subtle point here in the way the fusions are labeled with the M dash, the 5' X and 3' Y, is the dash between those two texts, which represents the fusion, is centered exactly below the fusion point, which reinforces the idea that the dash means the fusion. So the original diagram, the label with A, B, C, and X, Y, Z was so long that it actually had to scroll, or it had to wrap. And there were two dashes, notice. There was the dash between the A, B, C and the X, Y, Z, which was the fusion, and then there was the dash between the X, Y, Z and the word fusion, which is a different kind of dash. It doesn't mean fusion. It's a fusion of words more than a fusion of genes. And all of those points, even if they are maybe, to you, sound a little pedantic and a little bit, OCD-ish, that is where clarity comes from. Clarity comes from somebody else worrying about all the words that they use and only using the ones that they deem important and attentively throwing out all the other ones. Somebody was OCD enough and anally retentive enough to worry about every single detail so that you wouldn't have to. And that is your goal as a speaker and as a scientist and as an explainer, is to be that person, up to your capacity and emotional stamina. Here's an image which talks about a really neat concept called the DNA book, and half of it is spent telling you how you can get it and where it's made. And that is not the innovation here. Innovation here isn't a new mode of delivery of things from point A to point B, but it's the thing. And if you look at it, and the less you are in biology, the better, actually. Can you possibly imagine what this is about? It's a book. It's a DNA book. I'm going to guess there's some DNA in the book, because it's the elements, even for a biologist, are pretty confusing. Here's a redesign that tries to capture the essence of this book, and it shows only the thing that is the innovation and only the things that are important to capture what this book is about. And it doesn't distract you with anything else. So this idea that you could remove a page from the book is important. So it's some kind of like a 3-ring binder or something you can pop out. That's important. Then this idea of being able to punch out an element of the page or cut it out, that's important. And then this idea that this piece of paper you punched out, if you put it in a tube, which is the metaphor for the experiment, whatever the experiment might be, the thing that was on the piece of paper goes into solution, or actually becomes a reactive reagent, which is really cool, right? It's like having spices for your food embedded on pieces of paper that's biodegradable. And if you just want a combination of spices that captures the flavors of a country, you just tear this page out, put the page in the soup, and the page dissolves and you get all the spices, and you don't have to worry about the proportions. It's actually really cool. It's sometimes hard to know what is the round cow approximation. What do I keep? What do I throw out? And invariable people make the mistake of keeping too much, and then making a complicated explanation from fundamentally a simple concept. This slide is a complicated slide. There's way too much going on, and after 5 seconds of looking at it, I still don't know what it's about. And that is about the cutoff point. If between 5 and 10 seconds you don't know what's going on, then you're not going to be doing the work as the audience to try to figure out what's going on. You don't even know where to start. And one of the reasons is that there's lots of colors, there's a lot of directions your eye is being asked to flow along. There's elements whose purpose is unclear, like the pluses on top of the arrows. And as soon as you come across an element whose purpose is unclear, and if you do some work, it becomes like a bit of a detective story. You try to figure out, is this meaningful evidence, or is this not meaningful evidence? If you discover it's not meaningful, then everything else is cast into doubt. You start second-guessing what is being drawn and whether people care about how you feel as an audience member and so on, and then it gets messy. So the idea is, can I remove everything that's not important and make as simple an explanation as possible of what is important? Okay, it's got to do about networks. Okay, let's draw a network. Do I need a network of this many nodes, however many there are, 20 or whatever? Well, no, unless my network is indeed 20 nodes. But of course there's not. Networks are thousands of nodes. Biology is complicated. So why 20? Why not 2? Well, 2 is probably not enough to suggest a network or to show some of the behaviors of the nodes and classification of the nodes you want to show. So what is the minimum number that will capture the idea of a network, show people what you're doing, and have no more nodes than that? Well, you could try to experiment. I think 9 for this case. And I think in 5 seconds or 10 seconds, you know what's going on, or at least you have a sense that there's some orange stuff that gets identified and singled out. And at the beginning, I have stuff that's not orange. Then I identify some stuff that's orange. And at the end, all I have is orange stuff. And that's a great first pass. And you pretty much already know what this is about. And then in the middle somewhere, there's a step where I draw these boxes around nodes. And it seems like I only keep the orange stuff that was in a box. That's essentially the pipeline. I drop in the text to identify what the steps are. And then I drop in some more text to clarify what each of the steps does. But I do this in a way that doesn't get in the way of the main point, the main visual point. That flow of networks still has priority. And I leave it there. And I have only one color on the entire slide. And that color is that seed list, the gene list. That's very important. So I kind of see how what is on that list, how that is propagated across all the different steps. Now, I'll speak for just a little bit more, and then we'll take a 15-minute break. Sorry, I totally missed a question. Let's back up on slide 42. So on slide 42, the labels are now all in caps. Is this something you'd recommend? Okay, good question. Caps or no caps? When the caps are set in like a light typeface, but not bolded caps, then I think they might be a little bit easier to read, and they sit a little bit heavier on the page, and they provide a better label for things. As a top-level element, I think it works well. If there's not too many words, if the labels are short. And that is a aesthetic perspective on my part. And all the other elements, all the other labels are lowercase. So the thing here with this diagram is that there's several tiers of labels. There is the transformation process, which is the title of the image. Then there's the environmental signals, non-competent cell, competent cell, transformed cell. Those are the things. There is ddNA and so on. Those are all the elements. Those shouldn't be caps, because I feel like that would be imposing a little bit too much emphasis on individual elements, and you can't really emphasize all the elements, otherwise there's no emphasis. The caps are what each step is, and I think that the format of that label should be slightly different than the label of all the different elements. To do that, you could apply color, which I think is a bad idea here because we're already using red to be salient, and you want to maintain the emphasis of red by not using other colors. You can make the labels bigger, and I think changing typeface size is really not ideal because it takes up more room and it introduces too much variability in the formatting. You can make the typeface bolder, or you can do caps, but not both. And I already have bold typeface for some of the enzymes, so I've decided with caps. And that is a bit of a, I guess I walked you through some of the thinking process for me. You may arrive at a different conclusion, but as long as you're thinking about it, this is a good idea. Sorry, I missed that question. I'll talk for 3 more minutes, and then we'll take a 15-minute break, or 10, or whatever is best. So there is this process phenomenon called pareidolia, where we see patterns in random stimuli, like this. And we do this because we're pattern-seeking creatures, because we've evolutionarily grown up fearing predators in the bush, right? And if we saw a bush and we thought there was a predator in there and we ran away, that was good because we got to live. And it doesn't matter if there's a predator there. But if we thought, no, that's just a bush and there was a predator there and we committed a type 2 error, we would be dead. And so that person's paranoia, or lack of, would be taken out of the gene pool. So the only people that are left are twitchy, paranoid pattern seekers, which is great because it really helps us when we're running away from predators in the bush. And it's really bad when we're doing data viz. At the same time, sometimes, you know, maybe you don't have enough coffee, or you're tired, or you just don't see stuff because it's hidden. In this case, this is the predator's approach of trying to counteract your paranoia and your pattern-seeking skills. So when you combine the two together and you show figures like this, you stand the chance of triggering both. I don't know what's going on, so I'm just going to find some pattern that I can home in on, and I don't even know if that's an important pattern because maybe it's not. Or I haven't had enough coffee, and I spot the thing that I'm supposed to be looking at, either because I'm distracted by something else or because it's too subtle. So as the communicator, you have to lead the person towards productive interpretations of the data, because the data won't speak for themselves. They won't. I know it's a really attractive proposition, because if they do, then we don't have to speak for them. Then we don't have to speak. Then I'm done. It essentially abrogates responsibility. If I just throw enough facts at you, you'll get it. If I just throw enough data at you, you'll get it. You won't, and you won't. So here's 10 numbers. Here's some data. Do they speak? Well, what are the patterns? Well, there's lots of different patterns. What are they? Well, maybe some of you noticed that the first row is odd and the second row is even. Great, congratulations. Is that meaningful? I don't know. Do you know? You don't know unless you know what the author intended or what the data are about. You might say, well, I found some pattern and I'm just going to go with that. Depending on how the numbers are arranged, you might see that row dominant interpretation more clearly. But if I help you and I discretize the sizes of the numbers, I'm already telling you that the actual numbers themselves aren't important, but it's their relative size within a certain group, small, medium, large. So I'm already communicating to you things that I think you should be paying attention to. If I start piling the numbers up like this, I suggest that counting the number of elements within each group is important. And then if I really don't want you to worry about the numbers themselves, but just the counts, I would draw that. And that as a graphic is everything is necessary. Everything is necessary and sufficient to say that there's a significantly smaller number of medium-sized numbers in this set, however that was determined. And if I were to remove any of those elements in that graphic would be lessened, and the message would be incomplete. What I've done here is that my goal isn't to hide the numbers from you. You could download them as supplementary information and play with them to your heart's content. What I've done as a first pass explanation is focused you in on the things that I think, let's say as the author of this publication, are important. And as the reader, you should always remain skeptical, but generous to me that I've done my due diligence also, and that I'm not trying to hoodwink anyone, and I'm not trying to hide inconvenient truths. As some people invariably bring up this point of, well, you know, if you're controlling for detail, are you manipulating the audience? Not manipulating the audience. You're leading them to an understanding at a pace that they're capable of going, or that you're capable of going yourself as an explainer, but that they're willing to go at that rate. And you will get them to the end. It's just that you can't jump there right away. Don't overestimate your audience's ability to spot patterns in even simple representations. In this case, I don't know if you spotted it. If I was counting on you to spot it, I think that I would have been overly optimistic. I don't think I would have spotted it myself. And at some point, it isn't that you can't spot it, it's just that in a realistic scenario in a presentation, you just give up, and then you wait for the next slide. In this case, what are the patterns? These are the movement patterns, I think, of C. elegans, or another worm, and the arrows indicate I think firings between particular neural circuits associated with that movement. There's only 5 nodes in this little network, and you can imagine that the order of how those nodes are placed might reveal or hide patterns that may or may not exist. So what order do you put those nodes in? Does the order that appears on the screen now, is it an intuitive order? The fact that reverse is on the right and forward is on the left, and dorsal and ventral don't seem to be in the positions that correspond to those terms. What if we rearrange them so that forward is more to the right, where we think forward should be? Reverse is this idea of backing up from forward, slowing in the middle, dorsal, top, ventral, bottom. And once I rearrange the labels, you realize that, wow, this is a very symmetrical pattern of nodes and edges. And you wouldn't have seen that symmetry in the original image. And it's up to you to try to explore your data. And I know it's very hard when the data set is really big. I mean, in this case, there was 5 nodes, and the person was surprised when I rearranged them. So this idea, again, that the audience will sort it out, that the data speak for themselves and the patterns will be clear to see, I think is false. There can be some simple patterns that get hidden because of changes in scale. So here you have an example where you have some line traces on the left, and at least 3 of them span quite a broad range of numbers, And then there's a whole bunch of traces at the bottom that are squished because you can't see them because the scale goes up to 45. So they're shown on the right with like a little cutout to try to focus on the traces where the number of variant reads is small. All the while, each of the traces is individually color-coded and shape-coded. And it just starts to get not only very messy, but the patterns are hard to see because you're splitting them up between the variant traces of the highly variant cases and the cases that don't vary very much. And also the patterns are hard to see because there's such a occlusion of color and shape. I think in this case is one of these situations where the legend is actually not necessary and it gets in the way. If you can arrange so that the labels are next to your traces, then you can completely forego the legend. And if you can arrange it so that there is not that many traces, or if the traces do not overlap each other too much, or there's not so many of them that you can get away with just grays or maybe a small number of colors, then I think that the plot can be more effective. What I've done here is I did not try to show the weakly variant traces together with the highly variant traces because you can't if you were to, in any kind of scaling. So I split them up into two different plots, and this has given me the opportunity to reduce the number of traces in a given plot, which means I have more room for labels. I can reuse some of the labels, like the y-axis label. And because there's 3 relapses, I can label them 1, 2, and n, or n relapse, and use the word relapse only once. There's other ways that you can think about doing this if it's important to draw attention to how the variant count changes as a function of relapse. When all the patterns are overlapping, if they fall into certain families, or if they can be grouped by increase at the beginning, decrease towards the end, or increase towards the end, and so on, it's much harder to detect that. But if you split them up, like shown on the right, then it's a lot easier to see. You can kind of see that the fourth one, 241Y, is really quite different because it's the only one that is monotonically decreasing. And the last one is the only one that's monotonically increasing. And that's really easy to see in that plot, but it's much harder to see when you have all the other traces that compete for your attention. In genomics, at least, a lot of data, a lot of the results have to do with differences. And there's a lot of similarity between genomes, because that's what genomes are. They're mostly containers of differences. And we want to focus on those differences. And in a plot like this, where there are differences that they're there, you can see them because the letters change and the colors change, but there's so much similarity between individual lines that the differences get lost. And the extent to which each line is different from the others is impossible to detect. So how can this be fixed? You should, if at any point in time you have a baseline or a comparator or some kind of a consensus trace, string, number, or anything like that, you show that only once, and then you factor it out from all the individual plots, and then you only show the differences. So I cannot count on the reader's eye to spot all the differences. I have to help them do that. Now they can spot the differences in the differences, which is the second order difference that before was impossible to find because the differences themselves were very hard to find. I can reorder the lines based on how many accumulated changes they have. So this gives some reason to why they are in a particular order. And you can quickly see things, for example, like the shark, which is a fish, is very different in its differences to the next four, guppy, moonfish, ryefish, and trout, which are also fish which all seem to have the same kind of differences. You would not have been able to see that in the previous image because the shark is not even close to all the other organisms. So in this case, this is not a tiny data set, but it's not a big data set. It's a medium-sized, that's on the small end of medium, data set that you can actually manually explore and reorder these lines in a way that makes most sense to you and what it is that you're trying to communicate. You have to have a point. And the previous case, very difficult to make any point here. Lots of points to be made here. If you think back to those disordered sets of dots that I showed you before and how it was hard to count them, here's another principle that comes into play, which should be obvious what the principle is from the slide. So you should avoid grouping things unintentionally where possible. In other words, you should be thinking about, does the space between these elements force a grouping that is not relevant, or does it help a grouping that is relevant? In this case, you see rows of different shapes because the shapes that are similar are grouped in your mind or are put together into a group. And for me to get you to see columns of alternating circles and triangles, I could either do this, which is a really heavy-handed way of breaking that grouping, or I could just add a little bit of space. And as I add a lot of space, you now see columns because there's enough space to override the similarity of shape. But if I drop in tone, then immediately that spacing is not sufficient anymore. You now see a diagonal line of dark shapes because darkness, or in this case tone, is a very powerful powerful agent of grouping. If I drop in color, that overrides that diagonal line, and now all you see is a column. And then if I drop in something like a red, a splash of red, then that comes up even before. So first thing you see is that red point to the exclusion of everything else right away. So there's going to be certain graphical properties that stand out more than others. And then depending on how similar the shapes and or colors are, they will be grouped by your eye, and some similarity will be inferred. And that similarity better relate to the data. Otherwise, you're leading people down an unproductive road. Here's an example of where the choice of colors and shapes is counterproductive to the kind of comparisons you're trying to make. And it's always hard for me to interpret this figure because I see two things, and the thing I see is not the thing I'm supposed to see, so I always have to fight this instinct of wanting to compare the blue with the blue and the red with the red. So I want to compare the blue star with the blue circle and the red star with the red circle. Why? Because hue is a much more powerful grouping property than shape. I do not see two stars next to each other. I see a star on top of a circle. The reason why I don't want to be comparing the blue star with the blue circle is because if I look in the legend, I would be then comparing the controls across monkeys. So I'd be comparing the control of monkey H to control of monkey G, which is not what I'm supposed to be doing. I'm supposed to be comparing the control of monkey H to the inactivation of monkey H. And so I should be comparing blue circle to red circle and blue star to red star, which is really hard to do because of the color overpowers shape. So instead of having a legend like this, because I have two variables, monkey, G and H, control and activation, I now have a table, I now have a hierarchy. So I can cast my legend as a table to help you realize that there's two variables and there's all combinations of them. I can make different monkeys different colors, and I can make different conditions, different shapes. And because inactivation implies a lack of something or something less, I will make a hollow shape. And then I will adjust the spacing to confirm the fact that I'm supposed to be comparing shapes and not colors. But of course, in this case, both are compatible because I want to compare blue to blue and black to black because of the way that I have arranged the legend that now individual monkeys are different colors. There's lots of space here. This could be a little bit more compact probably. But think about this idea of spacing and this idea of grouping. You want to look at all the red error bars and somehow see them together because they form a group, because red is a powerful, or hue is a powerful grouping property. The problem is that all of the vertical axes are different, and so the relative sizes of all of these error bars cannot be compared together. Now, you might realize that, but as soon as you look at this, your eye doesn't realize it first. Your eye sees something, and it looks at the different sizes, like, oh, they're kind of smallish. But whether they are smallish or not really depends on the scale. So this kind of color to be used for uncertainty across different scales, I think is a really bad idea. The uncertainty is the property of the data point and should inherit its properties from the data point. Here is an example where color groups are created by your eye because of similar colors, but the groups don't mean anything. And in fact, the groups that should be created by your eye aren't because they have different colors. So the red stands out. There's some labels in C that are in red, but there are some elements in A that are in red, the P pass and D pass vertical lines. They're totally unrelated. Moreover, the same labels that are in red in C appear in black in B, breaking that connection between the fact that they're actually the same. And one of the things that's happening is, like, if you think back to that 5-panel figure that I rearranged originally, this is a 3-panel figure. But should it be a 3-panel figure, or should it be a 1-panel figure? Are all the panels here actually more closely related than would be suggested by the layout? Yes, they are. In fact, panels B and C should be on top of one another because that vertical bar that indicates the fold decrease in some variable is directly related to the experimental condition shown as an image of the gel. So it is, that bar is proportional to the extent to which that line is dim. And if you show it this way, then you connect the visual evidence of the data with the quantitative measure of the data, of the evidence. And they're complementary. Whereas if you show it like this, then you actually have to move your eye quite a distance before you look at the things that are supposed to be the same things. And if you look at the labels, control and knockdown, I believe, is KD. These labels repeat. But if I put minus and plus, that is enough to indicate control and experimental condition. Also, if you think back to this guideline or this process or rule, don't be too pedantic. If it is a fold decrease, it doesn't mean that the number has to be negative. So right now it says fold delta and dpass usage, because the delta is negative, the numbers are negative, and they go down instead of up, because that is how plots are drawn, right? Negative numbers go down, positive numbers go up, fine. But it's much more intuitive to have bars going up and being aligned at the bottom. And all I need to say is fold decrease in DPS usage. I don't have to make the number negative. I can make it positive by just saying, changing what it is that I'm talking about, and then it's obvious. So instead of having negative increase, I have a positive decrease. And I think that simplifies the notation and it simplifies the layout. Right, so as I mentioned, color is powerful when it is the only thing that appears, but everything is colorful, then the value of that individual color point is really diluted. So in this figure, there are a lot of different colors. And there's different categories or different parts of the image within which colors mean different things. So they're all categorical. In some of them, they're nominal categories, like the Coca 1 and Coca 2 and Lua. Those are just categories. They don't have an inherent order. Then there is kind of ordinal categories where there's mutation, gain, loss, so that implies a number underneath. And then mutation plus gain is green plus red, which is brown. And even though if you mix a green and red paint together, you might get a muddy brown, it doesn't mean that that is the color you should choose, because that brown is actually less salient than the green and the red. But it's more important because it contains both kinds of mutations. So whatever color you pick for that, it should indicate that there is more going on than in the original independent categories. And then there is some other category, kind of a heat map on the right. And red is used in different places to mean different things. If I isolate the red by making everything else gray, then the red really stands out. But because of the other colors, it doesn't have a chance to stand out. So it loses its value. And when it loses its value, you should realize that then I might as well not be using color at all, because there is very little value in it. Try to make decisions that give you the most return on your investment for using color. In this case, color is very powerful. But if everything else was colorful, then that color's value would be diluted. Color is a property of the data, or it is a way to say something about the data. But try not to have that color then spill into navigation elements around that data, because then it makes those navigation elements harder to see as a group. In this case, the strand axes take on the color of the data. That is completely unnecessary, because there is no way that I could confuse the two different y-axes, because they are physically connected to the x-axis on which the data sits. So there is really no need to do that. How would I address this plot to make the data stand out more? So again, as in the original panel, I would set up my axis in some way. Here you could fiddle with the spacing, and then I would drop data in, and that's it. Everything is black except for the data. And now that the only thing that is colored is the data, it actually draws attention to it a lot more. You can even argue here that you don't need to have color for the data because they are on different plots. And the identity of the trace is identified by the label on the left. So color here is being used redundantly, as was in the original figure. And here, if you look at carefully, the bars above the axis are darker than the bars below the axis to try to emphasize the difference between positive and negative strand. But that's something you get already. So you could, by the position of the data below or above the axis, so you get all of the formatting and all of the categorization of data for free by placement. You don't need color. So whether you use color or not here is up to you and what point you're making. But you should be aware of the fact that it's now redundant encoding here. And some people feel that if it's redundantly encoded, then it should not be redundantly encoded and the color should be removed. The choice of color is really important because it implies a particular direction in the comparison. So while this might make sense, control is gray and patient is orange, this makes much less sense that patient is gray and control is red. First of all, red is the color of emphasis. It's the color of disease. It's the color of things that go wrong. You should always reserve it for the worst case scenario. And in this case, what the plot is saying is compare the red to the gray. Something has decreased. But that's not the direction of comparison. We should be comparing the gray to the red. Something has increased, the variation count. Try to avoid color where you can and see whether you can have a successful figure. So a lighter gray, a darker gray for control and patient would work just as well and carry the same amount of information. Picking colors is hard because a lot of the ways in which we choose colors in software does not correspond to how we perceive them. We don't see colors in RGB. Our eyes do not even really get lightness and the concept of lightness and saturation. There are color spaces or ways to express color that are closer to how the eye perceives them. And some of these rules are captured in the so-called Brewer palettes, which fall into categories: qualitative, where the colors appear to have equal importance and don't have an implied order; sequential, where they do have an implied order; and the perceived difference between adjacent colors is similar; and diverging, where you have essentially two sequential palettes back to back. Some of them are colorblind safe, and you should always be thinking about people who cannot discriminate between red and green. That's the most common type of colorblindness. Even if you're just using grays, the gray tone Brewer palettes are very, very useful. If I had a metabolic pathway and I wanted to color the nodes differently with just gray, and I uniformly sampled gray levels, then you could see that in the middle there would be fairly poor tone separation between EGFR and GAPs, kind of similar grays, and then suddenly RAF, PKC, CK2, there's a really large tone difference because of the way that RGB mapping and our perceptions of tone differ. But with Brewer spacing, the differences in the grays along each of the steps is made more uniform. And so now RAF doesn't seem that much darker than PKC, which doesn't seem that much darker than CK2, and so the grays progress more evenly. So I encourage you to use Brewer palettes wherever possible. Lots of software supports that. Cases like this, where The colors were selected by the author, arbitrarily. I don't know, however. And they're not compatible together. The green is really bright, the red is really dark, the blue is very dark, there's another blue then, which is light. Then you think, are the two blues related? I don't know. There's a lot going on in this figure. I want to focus on the colors. If you can just go to the brewer palettes, let's say the set 2 brewer palettes, and just pick some colors. It doesn't matter that one of them is gray, because all you're trying to indicate is to emphasize the differences in the shapes and place the boundaries of the different, I think it's a phase diagram of some kind. And separate out the encoding and information about the boundaries or the color spaces away from the plot. This idea of dumping them into the plot may seem well-intentioned, but then you quickly realize that some of those shapes are too small to accommodate the data, so the data kind of sticks out, and then you realize that, well, my original plan of keeping the data close to the shapes cannot work. I lose more than I gain, so you move it out completely. Just like with colors, picking shapes is also hard, and unfortunately there isn't such a thing as brewer shapes. It's a lot harder to transform shapes into some kind of perceptual scale. That being said, I think that if you wanted to encode important and not important, and you picked a triangle and a circle, as opposed to a solid circle and a hollow circle, then one of those cases would make it much more difficult to encode somewhat important. What is somewhat important between a circle and a triangle? There is no readily guessable shape that goes in there. Whereas if I have a tone that I use to encode importance, then you can guess that maybe it's a lighter circle or it's a half-filled circle or whatever. This comes into play when you're drawing simplified representations of objects that have certain properties and behaviors that relate in some way. And you want the shapes to relate to those properties as close as possible. And you want the shapes to kind of embody the property. So if red is a cancer cell, okay, that's fine. I can go with that because red is the color of disease. That's much better than the green normal cell. Perfectly fine choice. Those two shapes and colors are fine. Then what is the cancer stem cell? You can't possibly guess that that would be purple because purple isn't worse than red. It's just some other color. Also, immune is a hexagon. That doesn't really follow from the fact that normal is a circle. So there are arbitrary choices that are being made by the author. And the idea is, how do you come up with some kind of a concise vocabulary for these properties that is consistent? So it's hard. If you think of cancer as the solid black circle, and you think, okay, cancer is solid black. So cancer stem cell is cancer plus something else, plus propagation, plus something more dangerous. So if you put these, make that cancer cell look more prickly, it looks much more dangerous. So you wouldn't want to pick that up in your hand. That's the point. The neoplastic cell, the cell where there's just a little cancer growing, kind of makes sense to put the little cancer cell shape inside that cell to indicate that it's filling up. Eventually, it'll become a cancer cell. So you could see that shape growing. And the immune cell having a thicker outline, meaning it's more difficult to invade or more difficult to penetrate. Now, you can come up with your own set of symbols, and that's actually a fun exercise, and your answer will be different than mine. And as long as you have some rational basis for all of those shapes, and you can show how one transforms into the other, then you would have been successful. When these shapes are used as glyphs for plots, keep in mind that there are some shapes that have strong visual boundaries because the shapes themselves are different. Remember that idea of grouping. And some shapes have very weak visual boundaries because they're too similar to be distinguished. Always choose shapes with strong visual boundaries. Otherwise, if you have some shapes with strong visual boundaries and some shapes with weak, the eye will make groups of shapes that are supposed to be different. And people will perceive different groups than you imply. This is a good example of that. It is a plot of genes, and they are categorized as either being processed or non-processed, so PS or NPS. And all the other genes are just little stars. And then if the gene is transcribed, if there's something other thing that's toggled on for that gene, then it's in a black circle. The trouble here is that PS genes, which are the red circles, are the same color as genes, which are the red stars. And in fact, the star is a much more complex shape than the circle. And it kind of takes your attention. And you've got these two red things that your eye groups together, and then you've got these blue things that are totally different. And so what you see is you see a sea of red with some blue, and then you see these black circles, and they form a group regardless of what is inside them. And again, we're kind of going by the general similarity. And the legend also doesn't emphasize the fact that there is a hierarchy of importance in the variables. So if you represented the legend as a table, you imply that all the other genes, because they're smaller, drawn with a smaller shape, are less important, and they're background information. The idea of transcribed as being on, as being a solid shape, makes sense. And processed versus non-processed, there's something more, so red versus black, perhaps. Then if you draw that plot with this recipe, then you can actually focus on all the individual shapes as different groups. If I say look at the background genes, the little black circles, you can see them as a group. You can see the hollow red circles as a group. You could see the hollow black circles as a group. You could see the solid circles as a group, and so on. So you're just by choosing more rational progression of how the shapes change across the categories, and you maintain this idea of grouping. I want to talk about error bars, and then I think we might wrap up. Error bars are sadly poorly understood by most people, even though they use them. Another set of columns that we run is the points of significance in nature methods, which talks about statistics. And there's two that are really important for people to read. One about error bars and what they mean. So for example, if you don't know the difference between what a standard deviation error bar is telling you and a standard error bar is telling you, then you should definitely read what it's telling you, because the next time you come to a plot and you see some error bars, you will know what to make of them. And you know what the error bars are telling you. Moreover, you'll know what they aren't telling you, or they aren't capable of telling you. And I think more people should use box plots, because they're a lot more expressive way to draw samples than just a single point. One of the things that if you stop doing one thing after this talk, or maybe you've never done it, which is great, but there's some fields of science where this is done almost exclusively, and it's almost like everybody's agreed to do this for some reason, is error bars on bars. And the reason why error bars on columns or bar plots or histograms don't work very well is because they encourage us into a false interpretation of proportion. If my baseline isn't 0, then I judge the size of the bar to the size of the error bar in a very different way. In that last case, it looks like the error bar is 5 times less than the size of the bar. But in the first case, like it's 30 times less than the size of the bar. So suddenly now, am I being led to interpret this in a different way? Now, you should know that you're not supposed to do that, but the eye wants to do that, and you want to minimize what the eye wants to do all the time. This should have been a scatter plot, in which case you do not compare at all that negative space that sits below the point to the size of the error bar. You are more likely to want to compare the relative positions of those points and the size of the error bar. You get into issues like this where if you have an access break and you have error bars, of which here you can only see half the error bar for some reason, and this is commonly drawn where you're only given half the error bar, and so you're not able to really make any kind of assessments between maybe where the error bars overlap or not. But in this case, because of the error access break, error bars on one side of the access break mean something totally different or are stretched. You'll get into cases like this where the person has remembered that they need to have an access that starts at 0, but then they have an access break that goes up to 0.95. Of course, then there's really no need for that access break. And then if you have a dark bar and you have a black error bar, you see only half, but you have a light bar, you see the full error bar. So already realize that this approach of drawing error bars on bars precludes you from using black bars, because then you can't see the error bar. It just gets bad. So here's a plot where I think can be easily fixed by first making the labels legible. So you flip it around, and suddenly now the labels are horizontal, and I can at least read them without cranking my neck. Then, if I change the data into scatter plots, this gives me the opportunity to restrict the scale. Sorry, the title is overlapping. Right. This gives me the opportunity to restrict the scale and to amplify the differences in the data given less space. That's a good idea. So this is something that I think you should try to do as much as possible. And I have 2 minutes. I'll talk about this table. Do not forget that the table is also your friend in drawing data. And the table is a container for information. A spreadsheet is a container that you can manipulate. But it's also a pretty powerful communicator of information. And sometimes a table is all you need. If you think about whether you need something more than a table, you can kind of try to draw a graph based on a table, and then think of ways in which you can scale the visual representation of the graph into something that is still visual in the way that there's still some maybe encoding of the data, but it doesn't depart too far from the table. So you can have, for example, just bars. You could start to remove the tick marks and the grids. You can move the numbers inside the bar. And now you have what is like a hybrid between a table and a tiny little plot to encourage judging proportions. And if you have 2 samples, for example, you can try to minimally draw them, where now it's, again, more like a table, but the numbers are moved around and there's a little bit of visual guidance to the changes between the samples. So you could see that 3 of the numbers drop and 1 goes up. And you could see that right away because I have connected the lines. You could judge those angles really, really easily, at least whether they're tilted down or tilted up, and one number has stayed the same. So you get that for free really quickly. And again, in this case, you could drop the grid line and you can drop the tick marks. I think I will end there. I'm happy to take any questions for a while, as long as people have them. Thank you for listening. I hope that people have learned something and been able to take away a couple of guidelines to either break or follow for their own work.